Anji sir, yes.
Okay.
Yes, we can.
Well, good evening all. The meeting number 2263 of Rotary Club Chandigarh Midtown is called to order. I hand over the meeting to our President, Rotarian Sanjay Bhatia. Over to you, Sanjay.
Thank you very much, Sergeant Past President R.K. Luther, and good evening to all. A special warm welcome to our guest speaker for this evening, Dr. Purnananda Guptasarma. Warm welcome to Dr. B.C. Gupta. Thank you, sir, for being part of this meeting this evening. I also welcome Kavita, Vanita, and
my brother-in-law, Hira Bhai sir.
And all the members, and I see some spouses also present. So warm welcome to all of you.
Thank you.
We would start this meeting with an invocation, and I would request Anne Gurinder Duaji to kindly present the invocation for this evening.
Thank you.
Yes, ma'am.
Professor Guptasarma, chief guest of today's function, President Sanjay Bhatia, Secretary Suchi, Mrs. Indu Luthra, and my dear Rotarian friends. Points to ponder for today's evening. We humans are constantly in doubts and live in fear most of the time. We live in our safe zone and do not want to get out of it. But the reality is that the moment we leave our safe zone, we move into the realms of our highest potential. Friends, be fearless like an eagle. As it flies higher and higher, it feels lighter and lighter. Here, I would like to quote Leonardo da Vinci, who said, "Once you have tasted flight, you will forever walk the earth with your eyes turned skywards. For there you have been, and there you will always long to return." So, dear friends, take the flight with the vision of a commander and build stories from your own life every day. One such fable, which left an incredible image in my mind, goes as follows. One day, the sun and a cave struck up a conversation. The sun had trouble understanding what dark meant, and the cave didn't quite get the hang of light and clear. So they decided to change places. The cave went up to the sun and said, "Ah, I see this beyond wonderful. Now come down and see where I have been living." The sun went to the cave and said, "Gee, I don't see any difference." When the sun went down, it took its light along, and even the darkest corners of the cave were illuminated. That's why the sun couldn't see any difference. One of the quotes which I recall at this time: "The enlightened ones can never be sent to hell or pushed into darkness. They carry their heavens on their shoulders all the time." We thought that heaven is a place where we are supposed to go. Perhaps it is an apt state of mind we were supposed to achieve. If you are full of darkness within, full of negativity, fear, and doubt, you become a cave unknowingly. It's a hell within, and no matter how much you accumulate, you still remain hollow. If you are illuminated like the sun, then the darkness of the cave won't matter. You could be in the worst of circumstances. You will still be able to find a blessing somewhere. You'll be carrying your heaven with you. One of the enlightened souls to whom we pay homage today is Dr. Luthra. He illuminated the lives of countless people through his good deeds and actions. We fondly remember him today. Now, over to our most committed President, Sanjay Bhatia, to carry on the meeting.
Thank you, Gurinder ma'am. This invocation was had a lot of motivation and inspiration for all of us. Thank you so much.
My pleasure.
As Gurinder ma'am said towards the closing part of our invocation, that we are dedicating this meeting on the death anniversary of our Dr. Jawahar Luthra, late Dr. Jawahar Luthra. And for this, I would now request our Secretary Shuchi to kindly play the video, following which we would have the Shraddhanjali by respected Dr. B.C. Gupta.
Yes, sir.
Thank you, Shuchi, for sharing these memories compiled by the family. May I now request Dr. B.C. Gupta to kindly pay the Shraddhanjali to Dr. Luthra, please.
Honorable President Sanjay Bhatia ji, the chief guest of the evening, Dr. Indu Luthra ji, fellow Rotarians, ladies and gentlemen, it is my proud privilege indeed to pay homage to that iconic personality, Dr. Jawahar Luthra, to whose memory this meeting has been dedicated. I have very high admiration and respect from the core of my heart for this gentleman who was a personification of total commitment dedication, sincerity. Take his profession. You have seen the photographs. The man was totally committed to his profession, whether it was undivided Punjab, later it was Moga in our Punjab, Delhi Irvin College, greatly influenced by his ancestors, his uncle, his dad, and all that. So Dr. Luthra turned out to be a great ophthalmologist. So you have seen the pictures. It is very, very clear. He was associated with American Academy of Ophthalmology. So starting from America, he was with American Society for Cataract and Refractive Surgery. So far as India is concerned, so he was always associated with the Ophthalmology Society of India. Then the North India Ophthalmology Society, which gave him the Lifetime Achievement Award. Then comes Chandigarh. He was president of the Chandigarh Ophthalmology Society. So he served with distinction at various army establishments, including the Command Hospital, Chandigarh. But after his premature retirement from service, Dr. Jawahar Luthra dedicated himself to the cause of the welfare of the society. He became a member of this club 1982. He was a participant, a very active participant in any program, in any camp, in any procedure, anything concerned with ophthalmology. Dr. Jawahar Luthra was there. So whether it was something in Gurudwara Ratwada Sahab, whether it was a school for blind, numerous other places, camps and all that. So the towering presence of Dr. Luthra could be felt. I fondly recall a personal encounter with Dr. Luthra. Dr. Vanita was the president of this club in 2000-2001. The club organized a 3-day camp in the polyclinic at Sector 45. On the second day, our great Rotary leader, Mr. Raja Sabu, and myself. So we went to the camp. We made a peep into the room where Dr. Luthra was performing his procedures. He just looked at us. Of course, he met us. He explained the things to us. But we got the feeling, with his typical posture and look, that we should not spend more time. We should leave him doing his work. He was totally immersed in his work. That I saw him at that age. I think he was close to 70 at that time. So the man was a picture of transparent sincerity, which I saw very clearly in his eyes and all that. So we are very glad that Dr. Luthra's legacy is being carried forward. Dr. Indu Luthra, his gracious wife, is the torch bearer of all the good things Dr. Luthra done. Then the children, I keep meeting them, Vanita ji, Kavita ji, they are all carrying forward the message that Dr. Luthra left. So not taking more of your time, I can speak at length about Dr. Luthra. Today, we must take a pledge that the kind of message, the kind of person, the kind of behavior which was being exhibited, that must be carried forward. And it is a good education, good inspiration, good motivation for us all. So with these words, I pay my homage to that great gentleman, Dr. Jawahar Luthra. Thank you very much.
Thank you so much, sir. Thank you, Dr. B.C. Gupta. very enlightening. And it's really nice that we have the whole family here. I see Kavita, I see Vanita also here. And in my few interactions with the family, I must say that everyone in the family, besides having a never say die attitude, they are so vibrant, they're so full of life. And why I say this, because I know where this comes from. That is what I saw in Dr. Jawahar Luthra till the last days. He used to keep coming to the meeting despite the difficulty of accessibility, and a lot of times he did use the wheelchair. But that is what I mean by seeing the josh that he had in him. I see that in the entire family, and these things definitely want and continue to inspire all of us. I know Indu ma'am would have been happy to organize eye checkup camp on this occasion, but considering the conditions right now, the least we can do is dedicate this meeting to Dr. Jawahar Luthra. Indu ma'am, would you like to say something before I call upon the introduction to the guest speaker? Or
Mom, would you like to say something? Mama?
I want to thank, am I mute?
No, I can hear you, ma'am.
Yeah. I want to thank Dr. B.C. Gupta, Shuchi, Sanjay ji so much. They are speaking so good for that. Thank you.
Kavita, Vanita would like to speak, either of you now, or would you like to?
Maybe at the end of the meeting. Thank you so much. Thank you.
Right, so you must stay on definitely.
Yes, sure, we will.
Thank you.
Sanjay, can I say a word?
Hanji ma'am, yes, I would be glad to hear members speaking, definitely, ma'am.
We are really very grateful to Dr. Indu Luthra. She's already given us 7,500 rupees, little more than $100 for a medical project in the club. That is her donation. I would like to announce it on this solemn occasion. Thank you, Induji. Thanks a lot. And in fact, we are planning to have a donation of dialysis machine, and hers is the first contribution towards this.
Thank you so much. This is nothing.
She has given the first donation for this project. Thank you so much, Induji. Thank you.
Thank you, Kanji. Indu ma'am, as Kanji said, this is the Shubh Aram. It is the first token money which comes with a lot of good luck for us, and because this project is a big challenge for us, considering what Kanji intends to do, and with very little support, I think, as of now from the district or from the foundation as of now. Every bit helps a lot, and this has given us the courage to go forward on this project. I would like to now invite Professor Dr. Dua for introducing our guest speaker for this evening.
Yes. Good evening, everybody. My dear Professor Dr. Guptasarma, Chief Guest for today's function, President Sanjay Bhatia, Secretary Shuchi, and my most lovable fellow Rotarian friends. It was in 2003 that somebody came to this club, Rotary Midtown 3080, our club, and gave a lecture on the latest Human Genome Project, which was published in that very year and that very month. Human Genome Project was to find out, to map all the genes which are present in you and me at different places. That brought a revolution to medical sciences, to molecular biology sciences. That person who came to our club was none else than today's speaker, again, and that is Dr. Gupt Sharma. This is his second visit to our club. Professor Dr. Purnananda Guptasarma was born in 1966 at Calcutta and is a professor, Department of Biological Sciences at Indian Institute of Science Education and Research, which is popularly called as ICER in Mohali. That is a knowledge city, we call it as, and Mohali. He had his MSc honors, biological sciences, and BE, that is a dual degree, which is given in BITS Pilani, that is Birla Institute of Technology and Science, Rajasthan. That was year 1988, and he had his PhD in 1993 from Center for Cellular and Molecular Biology, a national most prestigious institute of our country at Hyderabad, and Jawaharlal Nehru University, New Delhi. Had his postdoctoral studies as a Wellcome Trust Fellow from University of Cambridge. And by virtue of this common affiliation, both of us, he and me, are co-alumni from the same prestigious institute. His field of specializations are protein engineering and design, cell and molecular biology, molecular biophysics, molecular genetics, protein-protein interaction, and microbial enzymes. If it appears to be some technical words to you, that means he appears to be one of the veterans of biological sciences. Before joining ICER, he has been a scientist and later on a senior scientist at IMTech Chandigarh, which is another national CSIR prestigious institute in Chandigarh, where he was there up to 2010. As already elaborated earlier, at present he is at ICER Mohali, and has occupied the prestigious chairs of Dean, Research and Development and member governing body in the last 15 years of his stint at ICER. While ascending his academic ladder, Professor Gupt Sharma has won Indian National Science Academy, A.K. Bose Memorial, Indian National Science Academy medals, CSIR Young Scientist, Council of Scientific and Industrial Research, Evra Young Scientist, and A.V. Rao Foundation Awards. Professor Gupt Sharma is a polyglot. You'll be surprised to find that he is a polyglot and has mastered 6 languages, namely English, Hindi, Bengali, Telugu, Punjabi, and Urdu.
Oh, nice.
Dwells in a highly academic ambience at his comfortable nest with his wife, Professor Dr. Mani Luthra, who is a doctor at PGI, Chandigarh, and 2 daughters, namely Shivani, who is doing her PhD at Stanford University. She is doing probably PhD in artificial intelligence and robots. That's one of the coming up field in the field of sciences. While the younger one is a shining school student in Chandigarh only. My dear Rotarian friends, you know in today's assemblage, we are remembering our dearest Rotarian, Dr. Jawahar Luthra, who said a goodbye to this world 3 years back. Though he left physically on 31st January 2018, but as a Scottish poet, Campbell puts it, to live in the hearts we leave behind is never to die. And since we continue to nurture his magnetic aura all around us, we are of the firm conviction that he is always with us. The aroma of Dr. Jawahar Luthra is being kept perpetually alive by the untiring efforts of his Rotarian Ardhangani, Madam Indu Luthra, who continue to feed the garden that Dr. Jawahar cultivated with love and compassion so that it never stops to bloom and blossom. Ladies and gentlemen, put your hands together and listen to today's presentation entitled COVID-19, Mask, Vaccination and India from our chief guest, Professor Dr. Gupt Sharma, who incidentally also happens to be a family friend of Dr. Luthra's family. Over to Professor Guptasarma, please. Thank you.
Thank you very much, Professor Dua, for a very kind introduction. And thank you, Auntie, Mrs. Indu Luthra. It's wonderful to be able to speak today in memory of Uncle. My wife and I first met Jawahar Uncle and Indu Auntie in April 1997, and we've had the fortune and privilege of knowing them since then, and also knowing their entire family. So we are indeed family friends. Also, good evening to all Rotarians, in particular President Mr. Sanjay Bhatia, Secretary Shuchi ji, and others who were on screen, Dr. Gupta, Mrs. Dua, Karan ji, and others, and also a warm welcome to Kavita ji and Vanita ji. Today, I chose to speak about something that's very contextual and contemporary, which is COVID-19. So Shuchi ji has said that I could share the screen and operate the PowerPoint from here, so that's what I'm going to do. And as Professor Dua said, I'm going to talk about things that concern everybody: masks, vaccines, new strains, and also what's happening to India, and why India initially was predicted to be a disaster zone for COVID, but somehow seems to have escaped it. Of course, to understand COVID and to understand its relation to all of these subjects, it is necessary to talk a little bit about what the coronavirus is. So I will show you a few slides, but I'll say very few words about each of them. And I hope that you will take away from each slide only that which I say about each of them. So in this slide, what you see on the left is a schematic representing the coronavirus, where you can see that it looks like a ball with these spikes that emerge from it. What you see is a two-dimensional representation of a three-dimensional spherical object. The purple part consists of a lipid membrane, which is picked up from the cell within which the coronavirus replicates. And inside you see this dark brown spring-like or dark purple spring-like object, which is the nucleic acid, or in particular the ribonucleic acid, which constitutes the genome or genetic material of the virus. The virus, as you know, is, like all viruses, half living and half dead. That means that it cannot replicate and grow on its own, but when it enters a cell, then it can hijack all the machinery of the cell to grow. And for doing that, it has to first enter a cell and then replicate inside that cell, emerge from that cell, enter other cells, replicate inside those, and so on. So like a Trojan horse that grows in strength, the virus enters cell after cell, but the very first cell that it enters, you can see here in the right-hand side of the slide, where the virus has this lipid membrane, and when it approaches a cell, which also has a lipid membrane, the two lipid membranes fuse very much like two soap bubbles fuse. So when you have a small soap bubble approaching a large soap bubble, you may have seen that they form an even larger soap bubble. So that's the process that happens, as a result of which the ribonucleic acid, which is the genome or genetic material of the virus, enters the cell. Now, once it has entered the cell acts like a factory for the virus. So there are 9 stages through which the virus proliferates. There are technical terms describing each of these stages, but I shall try and deconstruct that for you. Essentially, what happens is that the virus fuses with the cell membrane and releases the genome inside the cell membrane. And then the genome itself is duplicated and multiplicated in a process called replication, because many copies of the virus are going to be produced, and each of them will need a copy of this genome or genetic material. So that is the first thing that the virus occupies itself with. And the next thing that the virus occupies itself with is to produce the few proteins that are present on the surface and for the packaging of the genome. So those are produced through this process called translation, and then using a part of the cell, which is called the endoplasmic reticulum, the viral proteins insert into the membrane, And then through a process of budding, which is the opposite of the process by which the virus comes and fuses with the cell membrane, the virus gets released in multiple copies back into the environment. So in this slide, you can see that there are 3 copies shown. That is just to signify that it enters as 1 copy and exits as many copies. Each of these then go and infect other cells so that in time, a large amount of virus can be produced. But even if a large amount of virus is produced over a period of weeks inside a human being, the total volume of the virus that is produced is not large. It can be compacted into a volume which is like the head of an alpine. So it's not really a large amount of virus. And so it's not the virus that's doing the damage directly, because we have a few 100 trillion cells in our body. And even if the virus exponentially goes up in numbers, it's going to take a long time for it to affect the body. So what is then the reason that the virus affects us? It is that it triggers a response inside the body in what is called the immune system. Now, there are 2 parts to this immune system that I would like to explain to you. The first involves this word that you would hear being bandied about in the news media all the time, the word antibodies. Antibodies are proteins, and I would like to tell you in a very few lines how they are produced, how they are generated, and how they work. So inside our bone marrow, there are cells called B cells. These B cells, as they grow and proliferate, rearrange a part of their genome to create a combinatorial diversity of many different types of what will eventually become antibodies, about 1 billion to 10 billion different types of antibodies. These are being produced blind. That means they are not being produced for any particular purpose. They are being produced all the time. But as you age, at any time, which specific combinations of the genetic material responsible for making antibodies is happening can vary from point to point. And so there are 1 billion to 10 billion antibodies. The hope of the system is that whenever there is an invader, then there will be something on the invader which can be recognized by 1 or 4 of these antibodies. So that's the process that happens here. The coronavirus interacts with 9 or maybe 10 billion naive B cells, which are virgin cells, which have not met any virus or any other invader. The protein on the virus, which is the spike protein, which is shown here in blue, which is also called the antigen, which is the opposite of the antibody, that is recognized by some naive B cells simply as a matter of probability, that if you have 1 billion to 10 billion different antibodies, some of them will recognize the virus. Thereafter, with some stimulation from the system in the form of the proteins called cytokines, this is another word which you may have heard bandied about a lot, cytokine storm. The B cell, which was a virgin cell, becomes activated, and then it proliferates, because what has happened is, from the large number of cells which could potentially have recognized different invaders, only the ones that actually do recognize the invader, they are activated to proliferate. And then when they proliferate, they do 2 things. One is they make copies of themselves and store them away as memory B cells, so that years and decades later, if you meet the same antigen and the same invader again, those memory B cells will come to life and they will mount an immune defense at the same time. So that's like a long-term stock or investment. The short-term one involves the cell actually undergoing this process called class switching and generating a large number of antibodies, which are called immunoglobulins, while they are on the surface of the cell, and antibodies once they get released. So there are many different types of immunoglobulins that get produced, and then there is a maturation of these immunoglobulins, a refinement of their ability to bind to the invader, and finally you get these plasma cells which secrete the antibodies. And the antibodies, what they do there is that they are like flag posts. Wherever a virus goes inside a cell and replicates, a little bit of the viral protein will appear on the cell membrane. The antibody will both go and bind to that cell and say to the rest of the system, hey, look, I put a flag here. There is an invader here, kill it. And also it will bind to the virus and neutralize the virus and neutralize its ability to enter a new cell. So the antibody works at 2 levels. it tells the system which cells to kill where the virus is working like a factory, and it also kills the product of the factory, which is coming out and being replicated. So this is how antibodies work, and the antibody levels actually vary like this. So as the antibodies go up, the virus which is proliferating, shown here in this purple curve, goes down, but the antibodies remain in the body for a period of a few months. In the case of COVID, they remain for a period of about 3 months, and the memory B cells and the T cells are there for a long time. But while all this is happening, there are cytokines being produced. There is inflammation, which is the result of the production of these cytokines and the killing of cells. And there is also killing of cells going on at the same time. And then there are these plasma cells which are generating B cells, and there are memory cells of 2 types right at the top right-hand corner, where you see there are not just B cells, but another type of cells called T cells, which help B cells to create antibodies. So there are T regulatory cells here, which regulate inflammation, and T helper cells, which help the B cells to create antibodies. So if you look at the whole picture, and I showed you what's here on the left side of the slide before also, in terms of the stages of the virus using the cell as a factory to make multiple copies of itself, there are different stages at which you can attack the virus. So on the right hand, you see some text where I've said that antiviral drugs mainly target the replication of the virus at stages 4, 5, 6, or 7 out of these 9 stages here. And one example of that is the drug remdesivir, which you've been hearing a lot about. Almost everyone who has COVID is being given remdesivir. The other thing is that what kills you is not the virus, but our body's response to the virus in terms of inflammation. Inflammation causes thrombosis inside microvasculature, inside organs, and that results in organ damage. And so it's necessary to bring down that inflammation. And one of the drugs being used to bring down the inflammation right now is dexamethasone, but there are others which have also been used. So for the last few months, management of COVID in hospitals has involved a combination of remdesivir and dexamethasone. One cuts down the amount of virus being produced, one cuts down the reaction to the virus. In addition, there are people who are making recombinant or protein engineered factory-made antibodies, just in case your body takes too long to create the antibodies to combat the virus. People are creating the antibody cocktails and delivering them so that you can have an immediate effect on the virus. And this is the Regeneron drug that the previous president of the United States received after very foolishly contracting COVID. But then he was given this cocktail of antibodies, and then that took care of the virus. And it's turning out that of all of these approaches, the most effective approach is the antibody cocktail, but it's also the most expensive to produce. And so very few people can get it. And so only somebody like the President of the United States could get it at that point with emergency authorization. Now they are trying to give it to other people too. The last approach is vaccines, which also involves antibodies. The only difference is, instead of making the antibodies elsewhere and giving them into your system, you try and trigger the system itself to generate the antibodies, which can then neutralize the viruses. And so you do that by exposing the body either to the virus itself, which is by contracting the disease, then you generate antibodies if you survive, and most people do, or by giving heat-killed or heat-inactivated virus so that the body can react to that and generate antibodies, or give a part of the virus in the form of a protein, in particular the spike protein, right? So different vaccines are doing different things, and the masks are also acting in a way like a vaccine. How? I shall explain. Meanwhile, in this slide, what I want to show you is that as our understanding of what the virus is and what it does increases, each of these green boxes actually signifies a different therapeutic approach to targeting the virus. So you can see some of the familiar names here and which part.
I think
Now, the two things that I want to mention at this point is that because there is no time to develop a new drug, people are taking existing drugs and testing them for their ability to target COVID. And these two examples that I've shown you here are actually from this very week. That means since Monday, in the scientific news media, these two things have appeared. Colchicine, which is a drug which is used to treat gout flaring up, has shown very strong anti-inflammatory action. It normally affects cell division. So people are trying colchicine, and this new drug, which is 30 times better than remdesivir, called tilidepsin on the left side here you see. It's also called Aplidin. It's used to treat multiple myeloma and lymphoblastic leukemia. It inhibits one of the factors that initiates this process called translation that converts the mRNA into protein, and it does it, it's 30 times more effective. So there are new drugs also coming up which are antivirals. In terms of the vaccines, you've heard names like Covaxin, Covishield, Moderna, Pfizer, and so on. So they actually target different aspects of the virus's exposure. The Covaxin, which is made by Bharat Biotech, is a heat inactivated virus. It's a conventional vaccine using the same principle that Jenner originally thought of. So you take the virus and you heat it for about half an hour at 65 degrees, and the virus degenerates, and the assumption is that it cannot reactivate by reassembling itself. So you take that degenerated or destroyed heat-killed virus and put it inside the body. Alternatively, because the main protein that elicits antibodies is the spike protein, you can organize to produce the spike protein in the body, or you can give the spike protein itself. So that's how these three other vaccines have been developed. Either you can give DNA that will go into the cell, deoxyribonucleic acid, which will go into the cell and make RNA, which will make the protein, or you can give RNA, in particular messenger RNA, or you can give the protein itself. So for giving the protein itself, no vaccine has yet been generated or developed, but people are working on it. But for mRNA vaccines, which can go inside the cell and give rise to the spike protein, Moderna and Pfizer have done it. And all over the West, Moderna and Pfizer vaccines are being given. The reason it's not being given in India is because it requires extraordinary refrigeration, minus 80 degrees centigrade, and it's difficult to maintain such a cold chain inside India. But these other vaccines which are being given in India are either the heat inactivated vaccine or something called an adenovirus-based vaccine, where a different virus, which is quite harmless, has been engineered to produce the spike protein. And so you put that virus in a replication deficient manner. That means the virus will be going inside your body and not proliferating, but the DNA will produce the spike protein. And the assumption is that the spike protein will show up on the cell surfaces and elicit antibodies. So these are the different vaccines. In India, you can take either Covaxin or Covishield. There are several concerns about the vaccines, and I will address them now. The first is that some people think that the virus causes, the vaccine causes changes in your DNA. This is a completely invalid concern. The only DNA vaccine that is being given is the adenovirus DNA vaccine. First of all, it is replication deficient. Secondly, it does not contain the genome of the coronavirus. The only bit of information that it carries is information for producing the spike protein, and that is what you want to do anyway, right? So there is nothing that is changing in your DNA. It's the DNA of the adenovirus which is producing the spike protein without interacting with your DNA. Some people are worried about changes in your RNA. Now, RNA is a very unstable material. It's here today, gone tomorrow, or in fact, for most pieces of RNA, the half-life is of the order of a few seconds and tens of seconds. So this is one of the reasons why the Moderna and Pfizer vaccines become useless if the cold chain of minus 80 degrees is not maintained and the virus is not given very quickly to people. They are giving the mRNA inside a lipid envelope which goes and fuses with cells. But if it spends some time outside, it's wasted. That mRNA in any case cannot go and change your cell's mRNA in any permanent manner. So that's also an invalid concern. There are no changes in your body cells because the cells which are going to make your spike protein to elicit antibodies are anyway going to be destroyed. But yes, there is one partly valid concern, which is a strong immune reaction, which is always there for any vaccine. You know, for any vaccine that you take, you can get a fever, you can get a swelling, you can get other side effects. If you are aged, if you have comorbidities, if you have a very strong pro-inflammatory status inside your body, then yes, if the spike protein gets made in large amounts when you take the vaccine in whichever form, right, whether as mRNA or as protein or as adenovirus DNA, you may have a strong immune reaction, and very few people will have a strong immune reaction. The number is very small, and therefore the probability is very low, but this possibility is there, which is why there are advisories about who should take and who should not take which type of vaccine, which should be followed very strictly. There is one more concern regarding Covaxin, which is if the production quality is not maintained, there is a possibility that all the virus that is being inactivated is not actually being inactivated, and some virus may survive, or if the inactivation is not complete, some virus may reassemble. We have heard cases where people have taken, for instance, oral polio vaccine and have gotten polio, but the cases are like 1 in 100,000 and so on. So this is also very low in probability. And at best, it's like having a very poor exposure to the coronavirus, which in any case is not a bad thing for most people because it can elicit antibody responses. So therefore, the reactivation of the virus, although it's a partly valid concern, is also not really a concern. The only concern out of these 5 is that you might have a strong immune reaction. Nobody can predict whether you will have a strong immune reaction. Therefore, the only way to deal with it is to actually take it and see. Basically, quality control during production of heat inactivated virus is an important concern. A little bit strong immune reaction is an important concern. For all other vaccines, even if there is poor quality control, you can be assured that this only reduces the efficacy of the vaccine, but it does not compromise your safety. That means cheez kaam nahi karega, par aapka koi hani bhi nahi karega. So therefore, I think it's quite safe to take vaccines. Go ahead and take vaccines. And watch out for any side effects. Be ready with a few anti-inflammatory drugs. But those with chronic inflammation and associated comorbidities may have unpredictable reactions depending on how they are built internally and how they have lived their life. A few words about masks as vaccines. I think this cartoon that I've put here is something that everybody understands. That is, there is a correct way of wearing a mask and wrong ways of wearing a mask, right? So what does the mask do? Why does it act as a vaccine when you're not actually giving spike protein? What it does is that a mask over the nose reduces the entry of the virus, right? Because the air that one breathes has passed through the cloth or the fiber. And also because if somebody sneezes or coughs or breathes virus out in your presence, then the virus does not go straight into your nose or your mouth, but it passes through cloth and a lot of it gets held back. Some of it goes in. The little bit that goes in exposes you to the virus like a vaccine and causes antibodies to be created inside you without giving you a florid infection, right? So it introduces the virus in very small doses inside your body. So this is how masks act as vaccines, and you must wear the mask over your nose and mouth, and if possible, especially if there are more virulent strains in your environment, you should wear 2 masks because that's double lowering of the probability that you will get enough virus inside your system for infection, and you will escape with getting essentially vaccinated. There are several mutations that are coming up around the world. There are new diseases, and this is because the virus has not yet achieved balance. You see, if a mutation in the virus, that means something that changes an amino acid on the spike protein and on other protein, if it ends up making humans too sick, then a patient gets isolated and the virus is not able to transmit from patient to patient. On the other hand, if the mutation does not make a patient sick at all, then the immune system of the patient overpowers the virus before the person begins to breathe out virus, and so again viral transmission suffers. So if you look at it from the viewpoint of the virus, it's not good for the virus either to make people too sick or to not make people sick at all. So since the RNA of the virus is mutating all the time, what we human beings are doing is acting like guinea pigs in which the strains of the virus that have mutations that allow the virus to make people sick but not too sick, those are the ones that are beginning to dominate populations. So recently you would have heard in UK and South Africa and Brazil, there are some variations, some new mutants that are that people are very concerned about. And yes, these are of concern, but we don't know whether they will be of concern to India or not. India itself is a conundrum, and the reason is obvious to all of us. You have to just go out into the marketplace, or as my wife and I, we had to go to Baddi for something 3 or 4 days ago, in fact last weekend. And when we drove into Baddi, we found that the two of us in the car were the only two people wearing masks. And there were crowded marketplaces and people all over, nobody was wearing a mask. And yet, India has escaped this disaster. So there are several possible reasons for this. One reason is diet. Indian diet has many anti-inflammatory components, spices, fiber, herbs, a very large proportion of vegetables. This causes the gut or the intestines of Indians to be anti-inflammatory, and therefore the virus comes in but does not trigger a very strong inflammatory reaction. This is one of the chief reasons, I believe, that Indians are escaping. Another is youth. We are dominated by youth, and the youth have very few comorbidities. Now, comorbidities and inflammation have a very strong connection because comorbidities develop due to inflammation. Diabetes, cancer, many of these things actually develop because of having chronic inflammation for a long time by not paying attention to what one is eating. So the youth haven't had enough time to spoil their systems, and so because we are dominated by youth, we are escaping. The third is lack of hygiene. This is something that will surprise some of you. If you are too hygienic, as you would have heard about relatives in America and in the West having a higher incidence of allergies than people in India, the human system is built in such a way that we need to keep getting exposed to microorganisms all the time to prime our system. So when we live in surroundings that are too hygienic, then what happens is those T helper cells which help antibody production, they switch from T helper class 1 to class 2, and that causes a change in your system which makes you much more prone to becoming to developing allergies and to develop inflammation more easily, both chronic inflammation and allergic inflammation. And so one reason that we are escaping as a nation may be that we are not as hygienic as all the other Western nations. Another very important thing could be that Indian homes and offices have much more natural ventilation. So the virus flows in and flows out without hanging around in the air for too long, right? Whereas most Western countries have poor ventilation. They have a lot of closed circuit air conditioning and so on. Plus, we also have much more childhood vaccination. And some of these vaccines, especially the BCG vaccine given for TB, is known to prime the immune system during early life in ways that seem to be able to help to defend against COVID. And the last reason could be that India has just been plain lucky in getting a strain of COVID-19 which is less virulent as well as less transmissible. So there are all these 6 reasons, and in some combination they are helping India to escape, whereas India was predicted to be a disaster. So this is a summary of the talk, but I will not go into the summary once again, except to say, in what I've said in the last line, that the best way to change yourself is to avoid an excess of sugar, excess of alcohol, excess of red meat, and other allergy-inducing substances which differ from person to person. You know what causes allergy in you. So if peanuts cause allergy or wheat causes allergy of any kind, avoid them, which cause chronic inflammation. And there seems to be a very strong connection between chronic inflammation and acute inflammation, which anyway develops when you have a virus. The two combine to give you hyperinflammation, which is what is called the cytokine storm, which is getting people into ICUs and killing people. So I think this is the last slide where I am ending with the analogy of the seed. Think of the virus as the seed of an unwanted weed, right? Think of the exposure to the virus as the sowing of the seed inside you. Think of your body and your gut health and inflammatory status as the soil in which the seed ends up taking root and proliferating. Think of the pollution around you as the groundwater or rain that helps the seed to be viable in your soil. In fact, pollution seems to have a very strong correlation. Where there is high pollution, the virus attaches itself to the particles hanging around the air and titrates and concentrates the virus so that when you breathe that air in, there is a greater chance that you're taking in enough virus to actually develop an infection. Think of hygiene as the temperature and humidity that turns the soil against you, right? Because too much hygiene is not good. Think of room ventilation as the breeze that blows winged seeds away from your soil. We've all seen seeds which have wings, right? So if there's a breeze, it doesn't land on a particular part of the earth. Think of the antiviral drugs and antibody cocktails as the weedicides that you end up using to take out the weed that develops from that unwanted seed. Think of the vaccines as the roasters of the weeds that make those seeds unviable. Think of masks as cobblestones, right, that prevent the sowing. So if you cover up the earth with cobblestones and then you sprinkle seeds, very few of those seeds will actually take root and grow, right? So the masks are doing that. And think of the anti-inflammatory medications as the soil balancers, like soil pH balancers or soil nutrient balancers. So each of these things is important in determining what happens to you when you meet the virus. But there are some things that you cannot help. For instance, exposure to the virus, despite wearing masks, at some point the mask is off and someone next to you is breathing the virus, you will get it, right? So the best thing that you can do in this analogy of the seed and the soil is, if you can't do anything about the quality of the seed, then you can do something about the quality of the soil. So make yourself less pro-inflammatory by attending to the health of your gut, by taking the things that are anti-inflammatory in terms of fiber, herbs, lowering sugar, lowering alcohol, lowering red meat, because these three are very well known to cause chronic gut inflammation. And then in a matter of weeks, you can change your body's inflammatory status in such a way that the soil of your body is no longer conducive to the growth of the virus. So that's the message that I have in this lower left half, which is that since we cannot always prevent the sowing or predict the outcomes of the sowing that happen despite our best efforts, the least we can do is to reduce the amount of seed sown by wearing the masks and ventilating rooms and preventing crowding, that is following social distancing, and also sanitization so that we don't pick it up from surfaces. We can also try to make the soil of our body less welcoming of the virus, either through vaccination or maybe or by making the body less prone to overreacting through anti-inflammatory guts and diet. And this last thing you can see is going to be useful whether you meet the virus or you meet the vaccine, because both can trigger inflammation in some people who are more susceptible. Thank you. I think with that, the talk is over. I'd be happy to take questions.
Very nice. Excellent.
Could I ask a question?
Yeah.
I was just wondering, like one of my classmates, she's a gynecologist in South Indian Andhra, and she has a daughter who's a doctor, junior doctor. And so one of her roommates got the disease while she didn't get any symptoms, and she hasn't got herself checked. But the point is that they're giving vaccines now over there in the hospital, and she wonders if she has enough antibodies, should she then go ahead and take the vaccine, or it's a waste, or maybe it's even like, the opposite effect?
So if the person we are talking about is young, I
Yes.
would say, which she is, right, roommate, she will not be harmed by taking the vaccine.
Mm-hmm.
On the other hand, if her exposure either did not create enough antibodies or the antibodies are not long-lived, there are cases of people who have had a second infection within 2 months, and in some cases within 4 or 5 months. And the second infection can also happen because the strain that was there in her roommate created antibodies, but those antibodies are not good against some other strain that she gets exposed to. So the current vaccines are good at giving you a much broader spectrum of immunity than exposure to a particular strain of the virus. Because attention has been paid to some of the regions where the mutations can occur. So the vaccines are giving immunity against not all the strains. For instance, the Brazilian strain, I think, and the South African strain are not fully protected by the current vaccines, but the UK strain seems to be. So the jury is still out on that. But in her case, since she's young, she should take it.
Okay.
She will not be harmed by taking it.
Exactly. Okay. Thanks.
Could I also say something, Professor Dua?
Yes, sir. Yes, sir. Please go ahead.
Dr. Guptasarma, could you enlighten us? We hear about herd immunity.
Yes.
So we read in the newspapers that Delhi is going to achieve it, that about more than 50% people, they have already acquired antibodies. Dharavi in Bombay has already achieved it. New York has achieved it. So could you tell us something about herd immunity?
Yes, certainly. So what happens is that when you have a large population and the virus is spreading, the virus is essentially doing a random walk through contacts between people. Now, if you increase the probability that the people being met by someone with an infection are already immune, then you increase the probability that they will be exposed to the virus, but they will not themselves transmit the virus because they have antibodies, and the antibodies will kill the virus as soon as it enters their system and not get a chance to proliferate and be breathed out even by an asymptomatic person. The person will still be asymptomatic, but will not be breathing out virus. So the way of increasing the probability that an infected person will not transmit to another is to increase the number of people around that person who have already been exposed and who have antibodies. So when more than usually 60 to 70% of a population has been exposed to a virus, either surviving or remaining asymptomatic, when 60 to 70% of a population has antibodies, then the rate of transmission of the virus is so poor that it is no longer viable. So this is called herd immunity, where the whole herd has, or a large part of the herd has developed immunity. And so those who have not yet met the virus also remain protected by sheer probability. But that is just a statistical argument. If you are the anecdote, so you see, I will differentiate between statistics and anecdote like this. Statistics shows that if you smoke, you can get lung cancer. Statistics shows that if you chew tobacco, you can get mouth cancer. My father smoked Charminar filterless for about 30 years. And he had chronic obstructive pulmonary disease in his mid-80s and a little bit of what seemed like emphysema, but he never developed lung cancer. Now, my father was the anecdote, whereas everyone else is the statistics. So what matters is whether you are part of the anecdote or part of the statistics. So herd immunity is protective as a statistical argument. Herd immunity is not protective as an anecdotal argument, because if you happen to be the unlucky fellow, right, who directly contacts someone who has an infection, then even if 90% of the people around that fellow have antibodies, you don't have the antibodies, you will contract the infection. So herd immunity is something that governments can take comfort in, but herd immunity is not something that individuals can take comfort in. There is no scope for relaxation of vigilance for individuals. You can't say, "Ke, ha, India mein herd immunity ho gaya, so caution thrown to the winds, I will go around without a mask." Yeah, there is a low probability, but what if you're a part of the statistic? It's just like you can run across a road 100 times without getting hit. And somebody runs across the road the very first time and gets hit, right? That person is a part of the anecdote.
Very
But
nice.
on one hand, the statistic says that 10 million people in this country are affected. That means only 1%.
Yes.
On the other hand, they say that more than 50% in Delhi alone are affected.
Yes.
It seems to be a contradiction.
This is a contradiction. You are absolutely right. I didn't touch on that because I was just explaining herd immunity. I don't know what to make of this argument. It seems like a contradiction in terms. How can, you see, Dharavi is something else, because Dharavi is a very small contained region, and it is unicultural and homogeneous in some way. Delhi is not. So you have on the one hand Lutyens Delhi, and you have all the NCR regions. You have parts where people are living cheek by jowl. You have parts where there's nobody for maybe 500 yards near you. So I don't know what to make of this, honestly. There are all kinds of people making all kinds of arguments, but you see, herd immunity is only something to feel comforted by if there is any scope for relaxation of vigilance nationwide. So no amount of talking about herd immunity in specific locations is actually of any use to anybody. So, yeah. This is what I feel. I said it a little strongly, but I sense that you are also on the same side, because you said that it is.
Dr. Sahab,
Oh, Dr. Vora.
my question is, Dr. I want to ask one question.
Yes.
We got vaccine yesterday at Rotary Hospital, Ambala. My question is, the vaccine will provide immunity for how much time, and whether it is effective for the mutant strain. We got it Covishield vaccine shot yesterday.
Yes. So since there are no instances of the Brazilian mutation or the South African mutation in India yet, we don't know, because the Covishield vaccine is very similar to the AstraZeneca vaccine used in collaboration with the Serum Institute in Pune. So the UK vaccine has also not been tested against the Brazilian mutations. But certainly the UK vaccine, the Moderna vaccine and the Pfizer vaccine seem to protect against the UK mutants, new variants. But you see, the equivalent of Covishield has not yet been released in UK. It has only been released in India in an emergency mode.
Right.
So it's very difficult to answer your question. One can only answer it in terms of probability. So I would say that in terms of time, the vaccine is also giving you immunity for 3 months if you don't take a booster shot. Right? Every vaccine, a booster shot, what it does is it improves the memory cell production. Also.
And after the booster dose,
so
for how much time?
After the booster dose, one would say for years, but one could be wrong. The plain answer is nobody really knows. So the honest answer is nobody really knows. Because you see, vaccines themselves actually take years in development. So we get a lot of information about how long the protection is, 5-10 years sometimes. And this vaccine, this entire set of vaccines has been rushed into production. So many of the details are not yet clear.
Thank you.
Yes.
Thank you, sir.
Dr. Vora, please. Dr. Vora has a question. Dr. Vora? Chuchi, can you put Dr.
Can you hear me?
Yes.
My question is that why so much scare has been created about this virus when there are ways to overcome it. And scare is one thing which reduces the immunity. And if your immunity goes low, you are bound to get this virus or any disease for that matter. I was a victim of this scare in the beginning. So what the best thing I did that I stopped looking at the TV. I stopped the mainstream channel. And then I also started diverting myself towards the prayers and kirtans and Gurbani and all that. So that really helped me. So the question is why so much of scare? Like, I am very grateful to you. You have not created any scare in your talk. On the contrary, you told us some very good things, nice tips. Why can't people at the national level or at the main media, they also bring out something good about how we can tackle this problem? And my second question is, the lockdown had affected the economy of the country, and I hope this lot is being said about India being #1 in production in the production of this vaccines, and I think India will be having nearly 60% share of market share in this vaccine market globally. So, what is your take on this?
Yes.
My 2 questions.
Thank you. All the questions are very nice and also very germane. So the first question was, why was there a scare? And the only answer that I can think of is because in Wuhan, pictures were coming out of people who were falling sick and dying very quickly, and sometimes dying within 5 or 6 days. And they were dying because the innate immune system generates a response to the virus, which is an inflammatory response, long before antibodies begin to get made. So when scientists and doctors realize that this is a virus which can kill you before you have a chance to develop antibodies, they got really scared. And as you know, hindsight is always 20/20. So, but foresight is not 20/20. So yes, in hindsight, I would say that the virus is more scary for some people and not so scary for the population, but it was not possible to predict this, and that is why the scare was generated. You are absolutely right that the economies of all countries and of the entire world has been shot through the chest, almost with a cannonball, with the lockdowns. But you know, nobody really knew what's going to happen. It did seem initially as if 3% to 4% was the fatality rate amongst people infected. Only now it's becoming clear that on a global level, it's much less than 1%, and in India, it's even lesser than that. But we didn't have all this information a priori.
Right.
So, yeah, so I think the correct answer for saying why there was a scare is because A, nobody knew, to B, there was no international cooperation, C, China was not divulging anything, D, America, you see, is the exact opposite of India. America has India has 4 times the population of America, but America has 3.5 times the number of deaths. So how does one explain this 11, 12-fold difference between India and America? Those 5 or 6 points that I made, differences in diet, differences in the youth composition, differences in ventilation, differences in hygiene, all of these could be responsible. But because America is like a bellwether for the world, and you know, American culture and American news dominates the world. So when America was having a hard time of it, America scared everybody. But there were some people in America who said there is no need to be scared. Maybe they were right about India, but they were not right about America, because in America right now, the number of people scheduled to die by March is 500,000, right? Or something like that. I think half a million people. Yeah.
Excellent.
So
My second question about
yeah,
India.
the second question was?
India is going to emerge as the largest manufacturer, and it may boost up the economy also, economy of India.
Yes. Yeah, I don't know if it will boost the economy of India, but it will certainly help the owner of Serum Institute of Pune, who already has 35 Porsches or something like that.
And Bill Gates has a lot of stake in that Pune Institute.
Yes, and that is a very good fodder for conspiracy theories, but one does not know. One does not know the truth or falsehood.
There's a lot of conspiracy theories going on, even as on today.
Yes.
And not only people in India are doing that, but the Western world is also are coming up with these theories.
Yes.
We shift to Miss Kavita, Mrs. Kavita.
Right.
Okay, your question, please, and after that,
Yeah,
I have
thank
one
you.
question, then we
Thank you,
start.
Professor.
Thank you. Hi, Dr. Guptasarma.
Hello, Naveen-ji. How are you?
How are you?
Good to see you.
I just wanted to ask you a stupid question,
No question is stupid, sir. No question is stupid.
See, you said that, non-hygienic conditions are actually proving detrimental to the spread of this thing.
Yes, yes.
Is it a good advice to tell the Western world not to be so hygienic now, so that, will it affect if they stop having a sense of hygiene now?
Yes, definitely, because the virus to ravage the whole of the US is going to take time. So the time span over which this changes is actually months. How do we know this? When an Indian family goes to live in the US, even if they go to live in a region where there is rampant hay fever,
yes,
in the first year, usually depending on when they arrive and when the hay fever season is, they don't get hay fever. But in the second year, they get it. So that means that going from India, where there is poor hygiene, they are not as prone to developing a hyperinflammation in their first year. So for a few months, they are protected. And then, because they are not being continuously exposed to microorganisms, they are shifting their T helper type and becoming prone, right? So if you now mirror this in time and look at it in exactly the opposite direction, one can argue that those who are compromised because of too much hygiene, if they relax their hygiene, then in a time scale of months, they will be better able to curb the inflammation.
Oh, that will be much cheaper than the vaccines, they're spending millions on.
Yes, but human beings are creatures of habit, and it's very difficult. You see, even in our own households, sometimes even within the same family, you find that between the wife and the husband, the understanding of the need for hygiene is very different, right? I mean, some people would prefer कि सब चीज झाड़ू पोछा 3 times a day, and another person would say हफ्ते में एक बार भी चलेगा. Actually, from the point of view of health, the हफ्ते में एक बार guy is overdoing it, but हफ्ते में 2 बार guy is probably right.
Yes, absolutely.
Whereas जो दिन में 3 बार झाड़ू पोछा करा रहा है, they are wrong, because they are actually curbing our exposure to microorganisms because of their habit.
Thank you for putting me off having a bath every day now. I'll be stricter with my non-hygienic routine more.
That's
yeah, this is actually.
May I ask a question?
Who is there? Who wants?
May I ask a question, please?
Please go ahead.
Please. I wish to ask a question.
Yes, sir, go ahead.
Your name, please.
I'm Chandu.
Oh, Chandu, sir. Please come.
What we have heard in the media is that one of the vaccines which has been developed is premature in the sense that complete human trials are not yet over.
Yes.
Isn't that rather risky? That vaccine?
Yes, you are right. In fact, virtually all of the vaccines have been released in a hurry for emergency use. Not just the ones in India. The difference with the ones in India is that all the data has not been released publicly initially. I'm not sure whether it's been released very recently, but it was not released publicly. Whereas in other countries, whatever data they had, they released it. But there is no doubt that every country has rushed this through. To get all the data takes years, and nobody has taken years. Some countries are saying that we can think of how the virus is going through the population as data. That is, instead of actually recruiting people to clinical trials, we've given vaccine and then we're seeing how they're behaving in the population. But that's a facetious argument. It does not hold too much water. So you are right, it is risky. Practically all of the medication is risky. In fact, dexamethasone, which is being given to everybody, under normal conditions, no doctor would prescribe so much dexamethasone to anybody, right? Because it's a steroid, it will bring down your immunity, it will do many things to you. Suppose you have a latent, shingles infection, then you may end up getting herpes. If you have a latent cancer which is in situ and contained, and you take too much dexamethasone, you may end up getting a flare-up of that cancer because your immunity is being compromised. So yes, you're absolutely right. It is risky. These are unusual times, and doctors and scientists are advocating the taking of risks. But yes, you are absolutely right that it is risky.
No.
Another thing I've heard is that there's a particular vaccine being imported by India, and that is not being given in India, and we are, I understand, exporting that to countries like Bangladesh and so on. Is that correct?
Oh, I've not heard this. I've not heard this. Professor Dua, have you heard this? I've not heard this.
No, I have not heard this, and we have a small question from Dr. Vora, the last one, and before that, I will have a small query also, please.
Please.
There is nothing like an imported vaccine being exported.
Yes, I've also not heard
this. I'm coming at with my third question.
Sir, be brief.
This is it is quite a mystery that lakhs of farmers around Delhi, and they were not observing any social distancing. They were not even wearing the mask. And it is really a mystery what type of virus is this. It like you said, hygiene. I think we are a very good country having poor hygiene, and that is a plus point for us. And I think like one of the audience said that we should. I think we should export unhygienic condition to countries like India, like USA. So, what about? Hello.
Hello. Yes.
Yeah. Please go ahead. Finish.
So my question is with regard to the farmers. Now, not even now when they are being even today they are asked to vacate the premises. They are still not using any mask, and they are even the press people who go to interview them, when they are interviewing them, they are also removing their masks and not maintaining social distance.
I see. So there are two possible answers. One is that these people have been exposed but are asymptomatic. The other is that none of them is really infectious at this point, and therefore the infection is not spreading. A third possibility is that amongst themselves, they have somehow managed to develop herd immunity, because Punjab ke Kisan, they have a strong constitution. So it's possible that their natural tendency to have inflammation is so poor that they are exposed to the virus in small quantities and actually using this whole opportunity as an opportunity to vaccinate themselves unwittingly. Ye bhi ho sakta hai. So one does not know the answer. India is a mystery. Why India did not go the way America went? In fact, there is data in America where people have congregated. Every single instance of unwise congregation without masks has led to a flare-up and transmission of the virus. Every single one, whether it is at political rallies or anywhere else. Why this is not happening in India is really a mystery, but it could be for one of those 6 reasons that
I think one of the reasons may be that they're coming from the villages where the hygiene level is very low.
Yes.
No.
I don't know what fraction of these farmers are real farmers and what fraction are farm managers, but real farmers are being exposed to microbes all the time.
Dr. Professor Guptasarma, it's very nice that you have covered every aspect from alpha to omega. But may I add to your one of the observation, and Dr. Vora's answered that one, that we are for one of the schools safe in USA as well as in India and UK also, that one of the reasons that the immunity in this part of the world is more that we, all of us, you agree that we have two genomes. One is the genome of Dr. Professor Guptasarma, and the second is the genome of the microbiota which you have inside your body, which is totally different at the subspecies level in India.
Yes, yes.
That is why they have taken out the samples from the poo of the patient, and they find that those who are resistant to COVID, their poo is totally different from that of them.
Yes. So this explanation, this explanation, Professor Dua, is actually wrapped into the explanation about the anti-inflammatory diet, because the microbiome of the intestines, which becomes the microbiome of the poo, is actually regulated by the food you eat.
Yeah.
So when you eat excess of sugar, when you drink excess of alcohol, or when you eat excess of red meat, then the microbes that proliferate in your intestine and dominate the population are pro-inflammatory. They cause severe inflammation of the intestine, they cause leakiness of the intestine, they cause IBD, IBS, and they can also end up creating diabetes, high blood pressure, cancer,
yes,
everything else.
So the one-line answer to Dr. Vora's answer is that their microbiota and our microbiota is totally different
yes,
from the rest.
Yes.
And
but if the Americans, Professor Dua, if the Americans, you know, the well-to-do Americans all take some salads, but the poor Americans take the worst cuts of beef.
Yes.
And there is very clear data that this is called the BAME data. So B stands for Black American, BA. So they are having a much worse time. And a possible hypothesis is that they are not taking enough salads, because the salads would create microbes in the gut that could be protective and which would combat the effects of taking too much alcohol and too much meat and too much sugar.
And the microbiota is also inherited from the mothers. They find those children who are cesarean, they are more susceptible to this because they bring less microbiota, gut microbiota in their tummies, and later on, if they are fed on the poo of the mother, then it will, the tablets will.
Yes, that's a condition called muconium, which happens sometimes in the womb also.
Yes.
Yes, yes.
That was a technical, so I thought of not discussing.
Yes, very right.
Very nicely you have covered from alpha to omega, everything. So many things are clear, except that advocating unhygienic condition, in already unhygienic condition, then if it becomes worse, then it may be the source of some.
Yes, unhygienic conditions may end up increasing TB.
That's what I was going to.
Yes. Yes.
So,
could I just add a small?
There's a balance. Yeah.
Could I just add to the American thing? My uncle, Dr. Chaman Luthra, who's Dr. Jawahar Luthra's younger brother, he lives in Yuma, Arizona. And in fact, he got the first dose of the corona vaccine. And then he goes and gets the infection, the COVID infection. And he was admitted in the emergency, and he actually got a cocktail of remdesivir and the monoclonal antibody infusion, and also dexamethasone, and hopefully is in the way to recovery. So I don't know how many antibodies is produced after the first vaccine, but he did get the disease quite bad.
Everyone is a bit different. Everyone is a bit different.
Correct. Professor Dua is absolutely right. Not only is everyone different, on an average, when you are elderly, your ability to produce antibodies is also quite compromised. This is one of the reasons why, herpes recurs in people who are in their 80s. They have successfully suppressed it all their life, and then the immunity drops when one is aged and it comes back. So it's possible that Chaman uncle did not generate as much antibody as younger people would. I think that's what Professor Dua is also saying.
Yeah, and very lighter that our species will be called as sooner or later. It's a lighter way of life. Homo sapiens moderna, Homo sapiens coxbina, COVID shield. All these will be subspecies of one of these vaccines. So, madam, what happened in your country? About 30 people died.
No, but that's absolutely wrong news, because in the CKM, which is a mixture of old people's and a sick people's home, on an everyday basis, out of the 500, 600 that they have, 30 people die on a daily basis. So this is 30 people dying over many weeks of getting corona vaccine. So it's nothing to do with the corona vaccine.
But
so
they were given this and they were all above it. But it didn't make any difference. But it sent the wrong signals to.
Yes, absolutely.
There's a lot of misreporting also.
Yeah.
And that creates more scare.
Even that about the farmer agitation. Thank you very much. And I'll pass it over to now the president. If there are no more questions, we really had a wonderful scientific session. I wish we have a recording which will be available tonight or tomorrow, and that is worth to be preserving, and that is worth to be shown into the classrooms also, as and when the university opens. And the good news is that university is opening next week.
I would like to inform the chief guest that this recording will be on the YouTube by tomorrow or at the most on Monday. You can have a look at on the YouTube.
I pass it to our president.
Thank you very much, Professor Dr. Dua. Very well said. I would like to now invite past President Pranab Mukherjee to kindly present the expression of gratitude for Dr. Guptasarma.
Good evening, everybody. Am I audible?
Yeah. Very well.
Well, thank you so much. I first like to, how do I put it, blame Dr. Guptasarma for teaching us so many new terms in the last 100 minutes that we got our minds full of antibodies. In fact, everybody.
Mind-boggling.
That's it. But anyway, it was a most informative talk. And initially I thought it would all, like go over the cuckoo's nest. But if Dr. Vora kind of puts it all down on print, which he will in our bulletin, then maybe after 2 or 3 readings, we will start to understand it better. Nevertheless, I just realized 2 things. One is that one of the reasons we Indians have not managed to get it is because we are full of not just antibodies, but anti-everybody. That's the way
You are Pallav, you are Pallav.
Yes.
Everybody knows I'm anti-everybody, so I have no problems with the virus. So that's it.
I think Dr. talk has increased our immunity also. And after reading it in the bulletin, it'll go further higher.
Well, I mean, like Dr. Dua reminded us, we first heard him in 2003. And since then, I mean, the antibodies against the lack of scientific knowledge has hit us all. And most of us have really picked up after that. And I've been reading a few books on genomes and stuff like that. And it is all, again, we can blame Dr. Guptasarma. Incidentally, as a last line, Dr. Guptasarma, do you realize that the number of syllables in your name are double that of Shashi Tharoor? And one more than his constituency, which is Thiruvananthapuram. That's 7 syllables. You got 8. Thank you so much. And we'll catch up on the entire thing, hopefully in our bulletin, which will be there in the next couple of days, and also on the YouTube. Thank you once again for enlightening us all and ensuring that we all get into the antibody, well, not everybody, but antibody mode. Thank you so much.
Thank you very much, Pallavji. And let me tell you that besides that, we have the death anniversary of Dr. Joa Luthra. We have the anniversary of the first patient falling sick of COVID. The first case of COVID was detected in India on 30th January 2000.
That's not a happy news.
That is the anniversary, the birth anniversary of COVID.
Dr. Guptasarma, you are still there online?
Yes, I'm still there.
Dr. I am an old Pilanian. I am a graduate of 1958-62 batch of engineering from Birla College of Engineering.
I see.
I wanted to inform you, I'm one of the 80+ wala, and I am a little bit scared of taking this vaccine right now.
Yes.
That's what I'm thinking.
Actually, if you're above 80, you must consult the doctor and consult the conditions.
You see, in the old days, there are many diseases connected with the age.
Yes.
A little bit of diabetes, little bit of hypertension, little bit of problem with the sleep. All these things are there.
But Biden is of your age almost, sir.
Pardon?
Biden is of your age. So you have to go for the comorbidity and not the age.
Biden is 78, sir.
So then those 2 years difference is nothing in India.
No, 75 se upar wala sab theek hai.
No, there is one thing, Professor Dua. I think it's something that we have all actually realized. Somehow Caucasians don't age at the same rate as Indians.
Very right, very right. Okay.
So a 75-year-old Caucasian is like a 65-year-old Indian.
No.
You are not. You are not Caucasian. Let me tell the members of this club that we are Australians. We are not. We are all Australians.
What? We are what, sir?
Australites, beta. Australite. Australia का word सुनो ना, Australites.
What is that?
No, please listen. We have
That's another scientific word.
We have 4 races, 4 subspecies. One is Caucasian, the Goras.
Yeah, the Goras.
Other is Negroes of Africa. Then we have these Indian and Australia, Australites, not Australian who are Caucasian from this one. And then,
So thank you very much, past President Pallavji, for that expression of gratitude. I would now like to invite Indu Luthra ma'am.
I want to thank you all. President Sanjay Bhatia, am I audible?
Yes.
President Sanjay Bhatia, Secretary Shuchi Thakur, honored guest, our chief guest, my Rotary and Inner Wheel family. I'm grateful to all of you for showering so much love and showing so much respect to me every year on my most emotional day, birthday of late Rotarian Dr. Jawahar Luthra, and remember him so fondly. This year, I'm thankful to President Sanjay Bhatia, dear Shuchi Thakur, Rotarian Nitin Kapoor, who made it happen. My greatest thanks to Professor Purnananda, who acceded to my request to be the speaker of the day. My gratitude to Professor Inderjeet Singh Ji, Dua Ji, Dr. B.C. Gupta Ji, Guninder Dua, Pallav Mukherjee, and all the Rotarians who put in their best to make this function a great success. Here I take the liberty to highlight some finer traits of Professor Purnananda. No doubt, Professor Guptasarma is a genius, proficient, great scientist, and an excellent speaker. But first of all, he is a remarkable human being. Here I would like to share with you his empathy, kindness, and humanitarian gesture. I cannot forget that night. How can I? When the nurse and I were helpless when Dr. Luthra breathed his last. I shared the sad news with Professor Mani Guptasarma. To my surprise, Professor Purnananda reached my residence at 2:30 a.m. all the way from Sector 39 in the biting cold. I truly felt that he was like an angel coming to my rescue. He pacified me, consoled me, comforted me, helped me keep Dr. Luthra's body in the PGI mortuary, and stayed with me till 7 a.m. All my blessings with him and with all my Rotary and Inner Wheel members who always show close proximity to me. Thank you so much.
Thank you.
Thank you, Auntie.
Thank you.
Thank you so much.
Thank you, ma'am, and thank you for sharing that wonderful piece, wonderful experience that you've had. We were just listening to Dr. Guptasarma, and you've added a completely new and different and a big dimension to this. A gratitude from all of us, Dr. Guptasarma.
Please don't say that. Let me also share a small anecdote. So in April 1997, my wife and I were looking for a daycare center for our elder daughter, who was then only a few months old. And someone told us about Auntie, and we went to their house and met Uncle and Auntie. And Auntie said, actually, we have just closed our daycare center, but usme kya hai, we'll just open it up again tomorrow. My wife's maiden name is Mani Luthra. So she said, oh, tum Luthra ho? Fir to tum hamari beti ho gayi. Chalo, aaj se tum hamari beti aur ye hamara damad. So the very next day, they reopened the Sanjeevani Daycare Center, and our daughter went to the daycare, and my wife was able to join PGI, initially as a senior demonstrator. Later, she had moved to Punjab University for a couple of years, and then she moved back to PGI as an assistant professor in 2001. So in a sense, I think our settling down in Chandigarh could not have happened without Auntie and Uncle and their gracious love. And for quite a while, until my in-laws came to Chandigarh later, and even after that, I've always looked upon them like parents or like in-laws, both, I mean, you could say. And I've received so much love, and it's difficult to forget Uncle, because I was listening to everyone speak about him. It's the same thing about him that strikes a chord in everyone. He was a marvelous human being, committed, empathetic, and loving. And I think he kind of encircled all of us in the sphere of his love. And we used to love going to their house. And I mean, we've sort of never lost touch over all these years since 1997. Yeah. Although sometimes we meet only once or twice a year, but even now, sometimes we attend Sanjeevani functions. Although our daughters have grown up long ago. Yeah.
Just 1 minute. I fail in my duty if I don't thank my friends who came all the way. Subhash, Rotarian Subhash Dheer and his wife. They came from Ambala, Rotary Club Ambala, to attend this meeting. And then Manjeet ji came from Indian Council of Social Welfare. I'm the member of that. She came to attend it. And then Mridu is Dr. Guptasarma's wife's sister. So, and then I'm grateful to my inner wheel friends, Rekha, Veena, Ranjana, Dr. Deepti, and Nandini, and my brother-in-law came from Delhi to attend the meeting, Hira Bhai Sahab. So I'm really grateful to you all who made this meeting a success. Thank you.
Thank you so much. Thank you to the complete Rotary family and the Inner Wheel family. Thank you so much for remembering Dad with so much of love always. We are really, really grateful as a family. Thank you so much.
Yes.
Thank you.
Thank you so much.
Thank you.
Rotary family, all inclusive family, and you are part of that one.
Absolutely. It's been such a wonderful experience, and it's been so nice to have all of you. It should be we who should be thanking all of you to have joined this, and and
I'm so
being as part of the
grateful
family.
to Mrs. Dua Maninder. She spoke so good prayers. I got really emotional.
After all,
Thank you, Mrs. Dua.
After all, she is my wife.
क्या बात है डॉक्टर साहब.
So thank you very much, Dr. Purnananda. We have 5 more minutes, and I would like to, before I just invite the secretary for the announcements, my personal thanks to you for a very simple, straightforward, and yet intellectual talk. You did talk of statistics and anecdotes. And when I saw the topic, it said COVID-19 masks, vaccines in India. The whole focus when I read the topic was going to vaccines, but you actually brought out so many aspects of this. As we say, common sense is not so common. You brought out such basic things about the masks, about the diet and such things. It was really very enriching and very interesting to listen to you. Do stay on, and I would like to invite Secretary Shuchi for the birthdays, anniversaries, and the announcements.
Thank you, President Sanjay. Before we announce the birthdays and anniversaries, we have 2 announcements. The club would like to thank past President Ashok Puri sir for his generous contribution of $30 for Project Astha, the cervical cancer vaccination project that's going on, and also rupees 1,000 he has donated for the Annapurna project for their grandson's birthday, Siddharth Puri Krishnan, who's right now in USA, and his birthday was on 21st January. Congratulations, sir, and thank you so much. Also, the club would like to thank past President Ajit Gulati ji and Beer ma'am for contributing 2,000 rupees on the occasion of their anniversary, which is on 4th of February, for the club projects. Thanks a lot, sirs. Coming to birthdays and anniversaries, we have the birthday of immediate past President Salil Dev Singh Bali on 3rd February, Rotary and Minakshi Sahore on 31st January. We have the wedding anniversaries today of Rotary and Nandita and Pradeep sir. Congratulations, Pradeep sir. I think I saw you today. Also, we have the wedding anniversary of Rotarian Kalpana and Rotarian Praveen Agarwal. 30 January, we have the anniversary of Rotarian Dilpreet and Rotarian Amanpreet Singh. 1st February, Rotarian Dr. Anurit Kaur and Rotarian Rajkaran Bhatti. On 2nd February, we have the anniversary of Rotarian Asha and Rotarian Rajiv Agarwal. 4th February, the wedding anniversary is shared by Beer Ma'am, Ajit sir, and Raspal and Kulvinder Singh ji. That's all from my end. And thank you, President Sanjay. Over to you.
Thank you, Shruti. Our best wishes to all the celebrating persons, members, and the anniversary couples this year also. With this, we come to the end of today's meeting, and I hereby adjourn this meeting with